Hey everyone, and welcome. Today we're diving into a really powerful upgrade in the world of scientific analysis. It's called HPTLC. And trust me, it's helping scientists get a much clearer picture of the complex world all around us, from the food we eat to the medicines we rely on.
But before we get into the tech, let's start with a really basic question. Imagine you've got this complex soup of stuff. Maybe it's a plant extract, a sample from a river, or a new drug. How in the world do you pull out just one single important ingredient from all that other noise? It's a fundamental challenge in science.
Well, for ages, the answer has been a brilliant technique called chromatography. The core idea is pretty simple, actually. You get a mixture to move across a surface, and because different parts of the mixture stick to that surface differently, they separate. It's kind of like a race where all the runners start together but finish at different spots. And this simple idea has been an absolute cornerstone of science for a long time.
And for many years, one of the most popular, go-to forms of this technique was the original workhorse of the lab, thin layer chromatography, or as everyone calls it, TLC.
So with TLC, you take a flat plate, usually glass, that's been coated with a thin layer of something like silica. You put a tiny spot of your mixture on the plate, stick it in a chamber with some solvents, and then you just watch as the solvent creeps up the plate, separating all the components as it goes. For its time, this was a huge deal. It was simple, it was fast, and it was super cheap to do. But science never stands still, right? As researchers started demanding more precision, more repeatable results, and actual hard numbers, the good old manual process of TLC, well, it started to show its age. The results were often just visual estimations, and it just didn't have the tight control needed for modern science.
And that is where our hero comes in. Let's talk about the upgrade that changed the game, high-performance thin-layer chromatography, or HPTLC.
So what exactly makes it high performance? Is it really that different? Oh yeah, it's a complete overhaul. We're talking about much higher quality plates with smaller, more uniform particles. Think of it like paving a bumpy dirt road into a super smooth racetrack for those chemicals. You get a much cleaner separation. But the real game changer, automation. From applying the sample with robotic precision to using a digital scanner to get incredibly accurate data. So, to really appreciate the massive leap forward that HPTLC represents, let's put these two techniques directly head-to-head. The main story here, the biggest takeaway, is this jump from a manual, hands-on process to a fully automated one. With the old TLC, you had way less control, and a lot of the time, you were just kind of eyeballing the results. With HPTLC, you get insane precision and real, quantitative data. It's the difference between guessing and knowing.
This chart really breaks it all down. I mean, look at this. The handling goes from totally manual to automated. The sample size isn't a guess anymore. It's precisely controlled. And the data, forget about manual calculations. It's all software-driven now. And get this, HPTLC can even analyze things that are literally invisible to the naked eye by using UV light. Plus, it hooks right up to a computer. It is a total system upgrade.
So, how does all this automation actually come together? Let's walk through the process and see how this new workflow creates such amazing, reproducible results. Okay, so it all starts with one of those top-quality HPTLC plates. Then, a computer-controlled applicator, not a human hand, puts the samples on the plate in these perfect, narrow little bands. After that, the plate is developed in a sealed, controlled chamber for the best separation possible. And finally, a digital scanner called a densitometer reads the plate, measures everything, and turns those separated spots into a digital graph with hard data. Every single step is optimized for accuracy. And the sensitivity we're talking about here is just wow. The ideal concentration for a sample is between 0.1 and 1 microgram per microliter. That's an unbelievably tiny amount of material, which means you can get clear, solid results even when you're looking for trace components. It's incredibly powerful. And this quote from the source material really just hits the nail on the head. It's not about just seeing separated spots on a plate anymore. The key phrase here is suitable for both the qualitative and quantitative analysis. That means HPLTC doesn't just show you what's there. It tells you how much is there. It turns a picture into real, reliable data. But here's where it gets really cool. This powerful technique doesn't have to work alone. In fact, one of its greatest strengths is that it's a fantastic team player. It can join forces with other powerful analytical methods to do some amazing things. I mean, just look at this lineup. HPTLC can be paired with heavy hitters like mass spectrometry, which is a tool that can identify the exact chemical structure of a substance. So think of it this way. HPTLC acts as the perfect prep tool. It does the hard work of separating a messy mixture into pure, clean components, and then it can pass those components along to another machine for a super deep dive analysis. That's modern science for you. It's all about teamwork.
And by teaming up like this, scientists can create these incredibly powerful workflows. They can use HPTLC to separate all the hundreds of different compounds in a rare plant, for example, and then use mass spectrometry to pinpoint and identify one specific compound that might have medicinal properties. This combination gives them a level of accuracy that was just unthinkable before.
Which brings us to a final, really exciting thought. HPLTC took a classic, reliable technique and basically supercharged it with automation and precision. So the big question is, with this incredible new power to see what we couldn't clearly see before, what hidden components, what secrets locked away in our food, our environment, and our medicines are we about to discover next?